we are going to discuss about fish vaccination. I'm sure that all of you are familiar with the term vaccine. So in this class, we are going to discuss about fish vaccination, different types of vaccines used in fishes, and the steps involved in fish vaccine production and vaccine delivery system. So moving on to the definition of vaccine. Vaccine, in a simple term, we can say that vaccine is a biological preparation that improves immunity to a particular disease. So, administering vaccine into a fish, the immunity of a fish is increased towards a particular disease. So, we can say that a vaccine typically contains an antigen or agent that resembles a disease causing microorganism, and it is often made from the weakened or killed form of the microbe. So weakened or killed form of the microbe which causing a disease is used for the preparation of that particular vaccine. Then concept of vaccination. The concept of vaccination is that it is a very easy, effective, preventive and protective method of fish from disease causing. Vaccination is a process by which protective immune response is induced in an animal by administration of vaccines. So improved immunity can be developed in a fish by administering vaccines. And once fish is stimulated by a particular vaccine, the B lymphocytes present in the fish body will activate by producing antibody cells, and it will remain sensitized and ready to respond to the agent should it ever gain entry into the fish body. And these are the types of vaccines, namely live vaccine, killed or unactivated vaccine, attenuated vaccine, toxoid vaccine, subunit vaccine, conjugate vaccine, DNA vaccine, and cellular fraction. Moving on to the first vaccine, that is live vaccine. Live vaccine is prepared from using live microorganisms. Using reduced virulence of the live microorganisms, live vaccines are prepared. Oral polio vaccine and vaccines for measles are good examples for live vaccine. Then second one is killed vaccine or inactivated vaccine. So in killed or inactivated vaccine, the virulence of the microorganism is destroyed by using heat, radioactivity, or by using any other methods. And this killed or inactivated form of microorganisms is used for preparing the vaccine. Then next one is attenuated vaccine. In this attenuated vaccine, live attenuated vaccines are used. Here the majority of the microorganisms are partially killed. Then toxoid vaccines are used against the bacteria that secretes toxins. Then next one is subunit vaccines. In the preparation of subunit vaccines, a small portion of the microbe is used for the preparation of vaccine, and later on the microbes used, the small portion of microbe used will give immunity to the entire organism. The next one is conjugate vaccine. In conjugate vaccine preparation, the poorly immunity provided polysaccharide coats of bacteria are linked with proteins. Then conjugate vaccines are produced in that manner. Then DNA vaccine. DNA vaccine, when a genes of a microbe's antibody is introduced into a body, some cells of the fish body will take up that, and the DNA instruct the cells to produce a antibody molecule. Then DNA vaccine is produced. The next one is cellular fraction. It is produced by extracting the cellular protein. And moving on to different steps involved in the vaccine production. As we mentioned earlier, a vaccine is a biological preparation by using the weakened forms of microbes that causing a particular disease. So the first step involved in the vaccine production is the selecting the strains of bacteria or microorganisms. The next one is growing the microorganisms. After growing the microorganisms, the isolation and purification of organism is taking place. After isolation and purification, the isolated and purified organisms are inactivated. The inactivated organisms then leads to the formulation of vaccine. Vaccine are formulated from these inactivated organisms. Then later on, the quality is checked, vaccine's quality is checked and lot released. Moving on to the vaccine delivery system. Mainly, there are three methods of delivering or administrating vaccine into a fish body. They are injection, immersion and oral. Among these three methods, the injection method of vaccination is considered as superior because by injecting following the method of injection, the vaccine will directly enter into the fish body. The second method is immersion. The method of immersion is mainly used in fish fries, and the immersion is carried out through dip vaccination, bath vaccination and by the method of spraying. And in dip vaccination, the fish fries are dipped in a highly concentrated vaccine solution for about 30 to 60 seconds. And in bath vaccination, compared to dip vaccination, bath vaccination will take a little more time and less concentrated vaccine solution is used in bath vaccination. And apart from these two, spraying is also used. And third method is oral method. And as we all know from the word itself, it is understood that by oral method, the vaccine is incorporated in the feed of the fish and it is given to the fish. That's all about the vaccination system. Thank you.